Okay, I'm now going to lecture to just a small part of the last PowerPoint for the protein synthesis section of this unit. This PowerPoint has quite a few embedded links to animations, so make sure you've downloaded the PowerPoints, you have access to that. So for example, this slide here, it's hyperlinked to a good animation that describes, some of the details that are found within here. So make sure you do that and have a look. Really, the PowerPoint is going to cover how genes are controlled in cells. The idea here is that most cells, if you focus on the human, most cells have the entire human genome within it. That's like 30,000 plus genes. It's not like all of those genes are expressed at the same time in every cell. Like a bone cell might express one protein, one gene, whereas another cell, like a nerve cell, would express different ones. So there has to be a level of control within the cell. Another reason being that, remember, these genes code for proteins. The cell might not want to have that protein expressed at a certain time, or its levels must be kept in a balance. Like you can't have too much of the protein or too little of the protein. So really, cells need to be able to turn on and off their genes. And that's really what gene regulation is, turning on and off genes. And a lot of where it is regulated is at the transcription level. Remember, transcription is taking the message that's in the genes of the DNA and turning it into a mRNA transcript, which then would move out to the cytoplasm and get translated. So we'll talk a little bit more about transcription factors. There are some genes that are on all the time, and those are called housekeeping genes. So I'm not going to spend a lot of time talking about how eukaryotes control their gene regulation. I'm going to spend a lot more time talking about how prokaryotes do, because it's easier, and it's what we learned, what scientists first investigated when they were trying to figure out how genes are controlled. So the first few slides, I'm not really going to talk about too much, other than tell you that it's a lot more complicated than what we looked at in the transcription PowerPoint. In the transcription PowerPoint, we saw an RNA polymerase binding to a promoter region, and then it moved along the coding region and transcribed the gene into mRNA. But as you can see with this image, it's a lot more complicated. There's a lot of other protein factors that come into play. And we're not going to really look at the details of these. This would be for university level genetics. So really the takeaway from this is that transcription is regulated by these transcription factors, which are proteins. And that's not quite as simple as what we made it out to be in the transcription PowerPoint. But I'm never going to ask you to draw these things out or to explain in detail the role of these different proteins in regulating transcription. So if you look at this animation here, it will explain quite a bit from this section of the PowerPoint. And I'm not really going to go over it in too much detail, other than let you know that or emphasize that there can be control at all these different levels. You can control gene expression before transcription, during transcription, after transcription, during translation, and even after translation. So pay attention to the animation and have a look at what's in the chart, and that's what you're responsible for. We're going to spend more time talking about something called operons. This is the way that bacterial cells control their gene expression. And we're going to use the example of a lac operon and the trip operon. Both of these are operons that an E. coli cell, an E. coli organism would utilize to control whether or not a gene is turned on and off. So we're going to have to try to explain this using examples. So it's important that you look at the animation first, and then, so if you haven't done that, stop the video now and look at that animation and then come back to it. Okay, so to explain how the lac operon works, this is the E. coli that lives in our digestive tract, and it uses lactose that's in the digestive tract. Lactose is a disaccharide. It uses it for energy, as an energy source. So by using lactose, it has to break it down first into glucose and galactose, and it needs an enzyme to do that, an enzyme called beta-galactosidase. So it's able to make that enzyme, That enzyme is a protein, obviously. And so it has genes that code for that protein. But it wouldn't really be efficient for that E. coli cell to produce the enzyme when there's no lactose around. It'd be a waste of energy. So it needs to control whether or not that gene is turned on. It needs to be able to control whether or not this enzyme will be produced through transcription and translation. And when it's not available, when lactose is not available, don't make the enzyme. And when it is available, make the enzyme. So it uses this negative regulation system to control the transcription of the beta-galactosidase. There are 3 genes for beta-galactosidase, and it's part of an operon. So what an operon is a cluster of genes that's under control of one promoter region and what's called an operator region. And an operator is a set of regulatory sequences of DNA to which some repressor protein can bind. Okay, so this might be a little bit complicated. Hopefully the animation helps to clear it up, but I'm going to use some images to explain it as well, some simplified images. So here you have the operon. This entire thing is considered an operon. And there's a promoter region where the RNA polymerase can bind. There's an operator region. And notice that the operator overlaps the promoter region. And then you have the genes that the promoter and operator control the transcription of. Further down here, which is not shown here but was shown in the animation, is the gene for the lac repressor protein. That gene is not a part of the operon. So it will be transcribed and translated, and you would get the lac repressor protein. So what happens is when there's no lactose around, this repressor protein is able to bind to the operator. You can see it fitting in with the shape. Fits in and binds to the operator region, and because of that overlap, it really is blocking the RNA polymerase. So when the repressor protein is bound, the RNA polymerase cannot bind to the promoter region. Therefore, it cannot move down along these genes and transcribe them into the mRNA, and therefore these genes are never translated into protein. So that's what happens when there's no lactose around. But if there is lactose around, now the bacterial cell wants to make these enzymes so it can break it down. So when there's enough lactose around, it acts as an inducer or a signal molecule. It binds to the repressor protein. And when it binds to the repressor protein, it changes the shape of the repressor protein, and no longer can that repressor protein bind to the operator region. Therefore, there's nothing blocking the RNA polymerase, and it can move along these genes and transcribe them to produce the enzymes that are needed to break down the lactose. So that's how the lac operon works. There's another operon that I want you to learn about called the trip operon. So if you haven't looked at this animation, then stop the video and take a look at this one, because this operon works a little bit different than the lac one does. The previous one, the lac one, is considered an inducible operon because of the idea that the lactose must bind to the repressor protein to change its shape to allow it to not bind to the operator region. It's going to be the opposite situation with this operon. So the E. coli needs amino acids. It needs amino acids to make protein. One amino acid is tryptophan. So it needs to be able to produce tryptophan if it cannot obtain it from the environment. If there's not enough tryptophan, it has the ability to make it because it has the genes that are used to make enzymes that will make the tryptophan. So the trip operon is a way to regulate that. I mean, if there's enough tryptophan around, what's the point of turning on these genes and having them make more tryptophan? So the operon is a way to control the production of tryptophan. Okay, so it's a similar diagram, except for this time you have 5 genes that are a part of the operon, and then of course you have the operator and promoter region, and you have a repressor protein. When there's no tryptophan around, the shape of the repressor protein is such that it can't bind to the operator region. So it's inactive right now. So if there's no tryptophan around, the RNA polymerase is able to bind the promoter region, and it can move down along the genes and transcribe, and then eventually those transcripts would be translated and you would be able to make the tryptophan. But when there is tryptophan around, the bacterial cell wants to shut off the production of more tryptophan, so by blocking these. So it binds, the tryptophan itself binds to the repressor protein and changes its shape. So it's called a